are live. Well, that is good. So hello, everyone, and welcome to the latest talk in the Worldwide Neuro series. We first of all absolutely have to thank Tim Fogels of Oxford, because he was absolutely instrumental in organizing this series of talks. So I am Philip Bartle of the Baden Lab at the University of Sussex, in the very sunny Sussex over here. So today we're having Herr Professor Dr. Tim Gollisch. I hope that I did not forget anything there, of the University of Göttingen. So he will talk for something like 45 to 50 minutes. And after that, we'll have a hopefully very salubrious discussion. So as Professor Gollisch is talking and enlightening us about retini, please post your questions in the YouTube chat section, right? So it will be monitored by our colleagues, and we'll have hopefully very good discussions of those afterwards. Right, so if you're not entirely familiar with our speaker today, we have done a thorough background check on him, and we know that he knows a thing or two about retini. So originally his background is in physics, which he studied, which he was a student of at the University of Heidelberg. And after that, that was followed by a PhD in biophysics at the Humboldt-Universität zu Berlin, followed by a very envious career path as a postdoctoral research fellow in Harvard, followed up by a group leader position at the Max Planck Institute for Neurobiology in Munich-Martinsried. And now, to the best of our knowledge, he still resides as a professor of sensory processing in the Retina at the University of Göttingen. So I hope that I haven't revealed too much, that he still has got something to talk about. So I give you Tim Gollisch. Tim, could you?
Okay, I guess, and hope you can hear me now. Thanks so much for the nice introduction. You're up to date. I'm still in Göttingen, doing sensory research on the retina, and I'll try to share my screen here. And meanwhile, I can thank you, Philip, as well as Tom, for this wonderful invitation opportunity. This is really exciting to be part of this. It's a great initiative. So also thanks to Tim Fogels for putting this all into motion. I'm very happy that I can join this here. Okay, so I'll be talking about linear and nonlinear encoding of natural stimuli in the retina. So my lab is interested in understanding, okay, let me just get a pointer here, in understanding how the visual information that enters the eye is translated by the retina into spiking activity by retinal ganglion cells, in particular what sort of processing and computations and function is conveyed in this correspondence between visual stimuli and spiking responses. And if you ask about function in the retina, the classical picture that you typically get if you look, for example, into a textbook is that ganglion cells have these famous center surround receptive fields. So for an on-center cell, for example, the cell is stimulated by light increases in the receptive field center and suppressed by light in the periphery, and vice versa for an off-cell function, which is captured by these center surround receptive fields. And by the way, I just see that the internet connection maybe is sometimes unstable here. I'm calling from a small village close to Göttingen. I hope someone will interrupt me if I freeze entirely, but otherwise I'll just continue. Okay, so in this classical view of retinal function, the cells are thought to act more or less as spatial filters, in the sense that they integrate the light that falls onto their receptive field in a linear fashion weighted by the strength and by the sign of the receptive field, with plus or minuses. And then the activity that the ganglion cell has represents whether a visual stimulus matches more or less this preferred contrast pattern that is given by a receptive field. And this idea has been highly successful for understanding retinal function. It explains a lot of things. For example, how cells in the retina, ganglion cells, respond to illumination with spots of different sizes. So you can see that when a spot gets larger, responses get decreased because of the surround. It is thought to enhance spatial contrast, which is a useful function because the most interesting aspects in visual scenes are often edges and contours. And this has been also formalized in a very famous and successful theory that states that the part of the function of the retina is to reduce the natural redundancy inherent in natural images, which has been used by Barlow as well as Attic and Redlich in order to understand the purpose of these centers around receptive fields. But at the same time, we also know that ganglion cells can be nonlinear, as shown, for example, in the famous work by Enroth, Kugel, and Robson. For example, seen when you present a reversing grating, as shown here, that goes back and forth, or that cells for different spatial frequencies of this grating may respond to one phase, to one reversal of this grating or the other reversal, but at a certain spot, when the grating, so to speak, straddles the receptive field, and the receptive field sees equal amounts of lightness and darkness, black and white, then some cells show cancellation of responses, so their activity stays flat. Here is shown as the firing rate, indicating linear integration because it's cancellation, and those cells are the famous X cells. And other cells at this point where brightness and darkness is balanced show responses to both reversals. And therefore, they don't cancel out the light intensity that hits the receptive field and are therefore considered to be nonlinear and are called Y cells. And this raises the question whether this apparent nonlinear spatial integration of light intensity over the receptive field is important for natural vision. So about natural stimuli, there are surprisingly few studies about this in the literature. And those that are there sort of show mixed results. There are some papers that emphasize that linear receptive fields or models that are based on linear receptive fields work fairly well across cells in the mouse retina, as well as some primate data, primarily from the lab of Sheila Nierenberg. And other papers have emphasized that there's more of a mixed behavior with good performance of linear receptive field models for some cells, but for other cells also failures. So there seems to be a cell type dependence in the primate retina, emphasized in particular by the labs of E.J. Cicuniski and Fred Ricci, and in the salamander as well in the work of Steve Backer, Cyril Ganglion, as well as our own work. So we here aim at investigating this a bit more thoroughly, and in particular trying to connect the performance of linear receptive field models and actual computations that are happening inside the receptive field for the mouse. But before I tell you how we do this exactly, let me tell you where the data comes from. So in my lab, we do multi-electrode array recordings of isolated retinas. They sit in a dish on top of a multi-electrode array with a number of small fine wires to a couple of thousands, depending on the array that we use. And at the same time, we stimulate the photoreceptors by shining a computer screen, focusing a computer screen onto the photoreceptors of the retina. We primarily use a mouse as well as salamander, axolotl salamander retina. And occasionally, we've also had a chance to record from primate retina from the marmoset. And because I will be showing you data from each, I will always put a small icon into the plot that indicates what animal we are talking at what point of time. Okay, so using this method, these extracellular recordings where you can get the spikes of individual ganglion cells, the spike times, a graduate student in the lab, Dimos Karamandis, has been analyzing how ganglion cells in the mouse retina respond to natural images. that are briefly flashed onto the retina. So here shown for 200 milliseconds, and there's a gray screen interval of 800 milliseconds in between images. And he focuses here on images rather than movies, because that's a more direct way of focusing on spatial integration that we are interested in here. And when he can, he uses a batch of images, 300 images in total, in a random sequence, and each image is repeated a couple of times, so you can collect the spike times that are elicited by a particular image and quantify the response of a cell to a particular image by counting the average number of spikes that are elicited during the stimulus. Okay, so how do you connect this now to whether the cell integrates linearly or non-linearly across its receptive field? The most typical approach is to use a model, and this is also what Demos has been doing. And the model is very simple. So he takes a given image, and then he converts, or he compares this to a filter that quantifies the receptive field. So this is the receptive field that we get from a different measurement. It's not so important how we get it. We do get it from a spatial temporal white noise stimulus, and then we compute the spike triggered average, extract the spatial component, and fit it with a difference of Gaussians. So there's a center part and also potentially a surround part. Often this is kind of weak, and here you can hardly see it. And this filter, then we apply it to the image that was shown to the cell in order to compute the activation. This is done very simply, just on a pixel by pixel fashion. We weigh the contrast values, so the deviations from mean gray in the image patch, by the strength of the filter, sum all these pixel contributions up, and get one number for each image, which we call the linear prediction. Essentially telling us how much luminance signal is there inside the receptive field for a given image. And then we do this for all 300 images in the batch and compare this to the actual response of the cell. And we may get a picture like this here for 300 images, how the linear prediction corresponds to the actual spike count that we measure. And if this is a nice relationship that we can fit, for example, by a simple nonlinear function, then we can say the simple model captures well how the cell responded to the batch of images based on this linear receptive field. And this model is none other than the famous and often used linear nonlinear LN model. So here we would say this LN model captures the responses quite nicely, and therefore this cell is more or less consistent with the linear receptive field. So here are some other examples. Cell 1 we've just seen. Cell 2 here has a downward sloping, not positive peak. And this cell then likes negative contrast inside the receptive field, so blackness, and it's therefore an off cell. Here you can also see that we can capture on-off cells which have this U-shaped nonlinearity. So these are all cells where the linear nonlinear model seems to be doing fine. But then we also have examples where it really fails, where there seems to be no good relationship between the linear prediction of the LN model and the actual spike count. Even if we allow the nonlinearity to do funky things, like having this kink here, which we have in there because we want to be flexible enough to capture all these different potential nonlinear relationships between the linear prediction and the actual spike count. We can then quantify this to obtain an overview by essentially computing the correlation between what we predict by the model and the actual measured response. And we do this here. With a bit more detail, for correlation, we use what's called a normalized correlation coefficient, which we've taken from the literature, that takes into account the variability of responses, the noise in responses, given that we only have a limited number of trials. So 1 here in this performance measure means that the model is as good in predicting responses as we can expect, given that the responses themselves are noisy from a limited number of trials. And also, I should mention, we do this with cross-validation. So we always leave out a number of images, fit the nonlinearity, and then do the prediction on the set of images around, and in the end, take the average normalized correlation coefficient over all folds. And what we then see here is that we have a bunch of cells whose performance is near 1, so where the linear nonlinear model with the linear receptive field is doing a good job. But then there's also this long tail here containing a lot of cells where the model is not doing so well, potentially a sign for nonlinear spatial integration under natural images. But how do we connect this now? How do we connect this model failure for these cells here to actual things that are happening inside the receptive field? So let me show you this here. So this is one of the cells where the linear nonlinear model apparently failed. And what we can do now is we can look at pairs of images, like taking these two data points where the linear prediction was very similar, almost the same. So there was the same luminance signal inside the receptive field for these two images. But if we look at the responses, for one of the images, we had a strong response, and for the other, we had a particularly or fairly weak response. And if you look at the images, you may already guess that this has to do with the structure that's inside the image, because here we have a lot of contours, a lot of white and black, and here we have a more homogeneous illumination of the receptive field. So then we can ask, does this difference in the spike count have anything to do with this, with the spatial structure here inside the receptive field? And to quantify this, we compute a measure of the spatial contrast inside the receptive field by computing the spike, the standard deviation of all the pixel contrast values weighted by the receptive field. And for this particular image, we get a certain value, 0.46. And for this image here, we get a smaller value because there's less contrast inside the receptive field. Okay, and then we can hypothesize that the difference in spike count, if the linear prediction is the same, has to do with this difference in spatial contrast inside the receptive field. And doing this systematically, we can pair all of our 300 images into 150 pairs that have approximately the same linear prediction. And what we then get is a picture like this. So here's, for example, a cell where the linear nonlinear model did not so well, and the cell was sensitive to spatial contrast, as you can see here in these examples. And if we do this over all pairs, then we see that there's a systematic relationship between the difference in spike count for each image pair and the spatial contrast inside the images for the image pair. So if the luminance signal is the same for two images, then for this cell, we almost always get a higher spike count if there was more spatial contrast inside the receptive field. Here's the same analysis for a cell that doesn't care so much about spatial contrast, where the linear nonlinear model has been doing well. We get the same, very similar spike count here for different cells, for different images, although they have different spatial contrast. And you see here a more or less flat relationship between spatial contrast difference and spike count difference for pairs of images. And then interestingly, we also find a third type of cell, a third type of response, where the sensitivity to spatial contrast is reversed. So here's a cell where when the luminance information inside the receptive field is the same, the cell actually responds more strongly if the receptive field is illuminated more homogeneously, as shown here. And in this analysis, you see that with luminance signals being equal for image pairs, you get stronger responses if the spatial contrast was weaker inside the receptive field. We can relate this sensitivity to spatial contrast by computing a measure of spatial contrast sensitivity, which in these plots is just the slope, the regression slope between the difference in contrast and the difference in spike count, and then relate this to how well the cells perform, or how well the LN model performs for different cells. And what we there see is that essentially we have a bunch of cells that seem to be more or less linear. They have good LN model performance as well as no sensitivity in these the spatial contrast sensitivity analysis. And then we get a group of cells that are essentially nonlinear. They have a not so good LN model performance, and they shows sensitivity to spatial contrast, like this cell here, with a large slope in the spatial contrast analysis. Okay, so here we now have a connection between sensitivity to spatial contrast and encoding of natural images. So if we have some cells that care about spatial contrast and others that don't care about spatial contrast, we should also be able to see this if we reduce the spatial contrast, for example, by blurring images. And here I show you responses of cells from the same three categories when we show original natural images as well as blurred versions. You see here, for example, the cell reduces its spike count considerably when the image is blurred. This cell doesn't really care much about blurring. It does the same as before. Whereas this cell, you see again that it prefers more homogeneous illumination, so it actually increases its spike count when the image is blurred. And this is a systematic effect that we see when we take a number, in this case 40 different images, either in their blurred version or in their original version. This cell decreases activity, this cell rather increases activity. So that confirms the sensitivity to spatial contrast. And just a note at the side, if we have here cells that decrease their activity with blur, and here we have cells that increase their activity with blur, that sort of suggests that if you look at the difference in activity of these two cells, this gives you interesting information about how well an image is actually in focus. You can imagine this if an image, for example, gets out of focus, then these cells will mark this by increasing their activity relative to these cells that decrease their activity. And who knows, maybe the brain is using such a signal in order to adjust focus or learn the right focus for objects at particular distances. In this analysis, we also looked a little bit at cell types, but let me show you this briefly. So in the multi-electrode array recordings, there are some cell types that we can identify fairly easily. And one of those groups is what we call IRS cells. We call them that way because we have previously shown that these are cells, a functional group of cells that are particularly sensitive to image recurrence. So if you show images here, gratings that of different identity, different spatial phases, then the cell responds particularly strongly when for a transition of images, when before the transition and after the transition, the same image was shown. So they like a recurrence of an image. That's an older story that I want to go into detail here. But for us, it just here means that we have a fairly easy way of identifying a particular group of cells, which, as far as we know, correspond to the so-called transient of alpha cells in the mouse retina. And for these cells, we find that typically the LN model is doing a fairly good job with a high performance measure under natural images. By contrast, we have also cells that seem to be more nonlinear with worse performance, for example, direction selective cells. Maybe not surprisingly, because we know that these cells do interesting nonlinear things with visual stimuli, and they show this also here in the performance of the LN model. And then there are cells that are in between, for example, orientation selective cells. Some of them have a fairly good LN model performance, others not so good. And actually, it turns out that when you look at the distribution of a performance measure of how well the LN model does for orientation selective cells, you see more or less a bimodal distribution. This becomes more pronounced if we separate the cells into on-type and off-type orientation selective cells. The on-type cells seem to be mostly linear in the sense that they have a good LN model performance, but the off cells really show a strong bimodal shape here. We went a bit further and tried to cluster these cells based on how well they do in the linear nonlinear model, as well as how well they do or how much contrast sensitivity they show in the previous analysis that I've shown you, as well as using some other measures. And it turns out that we can actually find more or less distinct clusters of off orientation selective cells. And then when we look at the preferred orientation of these cells, we find that if we take a pair of orientation selective cells that are in the same cluster, those cells tend to have a similar preferred orientation, with a difference in preferred orientation peaking near zero. Whereas if the two cells come from different clusters, so a more linear cell together with a more nonlinear cell, they tend to have different orientations, preferred orientations. So that at least indicates that there's some relationship between what subtype of orientation selective cell we have for what preferred orientation it is sensitive to, and whether the cell is more linear or more non-linear with a good or worse model performance. So let me summarize this very first part by saying that we have seen here that nonlinear spatial integration is indeed relevant under natural images for some or many ganglion cells in the mouse retina. That we've also found some cells where the nonlinearity acts in such a way that it prefers actually homogeneous image patches with little spatial contrast, and that this property of being linear or nonlinear appears to be cell type specific, as we've seen from the direction selective and image recurrence sensitive cells. So this now raises the question how we can capture these nonlinearities in models. And this gives me a second part of this talk, but I will be very brief on this because this is essentially work that has already been published, but it fits in here well as a reminder. So typically it's thought that the nonlinear spatial integration in ganglion cells comes from the fact that ganglion cells integrate nonlinearly over an array of presynaptic excitatory bipolar cells. And this nonlinearity is thought to come from the fact that bipolar cells have a nonlinear signal transmission in their transmitter release onto the ganglion cell. So that means that the ganglion cell receptive field can be subdivided into what's called subunits, which correspond then to individual bipolar cell inputs. And if we want to integrate nonlinear spatial integration into models of ganglion cells, a good or necessary step seems to be that we have to find out where these subunits of a particular ganglion cell, where they are. So we've devised a method a couple of years ago, spearheaded by a former postdoc in the lab, John Liu. And this method is based on using spatiotemporal white noise as a stimulus, and then extracting those stimuli, those images that trigger a spike, that are successful in activating a given ganglion cell. If you average all these images, you get a receptive field. That's the famous spike-triggered average. But if you do a different analysis, for example, or what we do is use a method that's called non-negative matrix factorization. So together we call it spike-triggered non-negative matrix factorization. You get a number of receptive field-like looking structures, small filters. And interestingly, some of them look like noise, or they turn out to be noise. Here shown in blue, but the others here shown in red, they have a nice localized structure that look like subunits. And if you fit these with small 2D Gaussians, then you see that they cover this receptive field quite nicely. And we take them to be the subunits of a given ganglion cell. Are these? I should mention that we're not the only ones interested in devising methods for finding subunits in ganglion cell receptive fields. There are interesting related approaches, for example, from the lab of E.J. Ciccioniski using spike-triggered clustering, or from the labs of Steve Backus and Surya Ganguly fitting layered neural networks to ganglion cell responses. And they also get similar layouts of subunits inside receptive fields of ganglion cells. So for us, we asked then, are these subunits really bipolar cells? Do they correspond to individual bipolar cells? And Helene Schreier in the lab analyzed this by heroic experiments where she recorded with sharp microelectrodes from individual bipolar cells, while at the same time recording spikes from ganglion cells, so that she could get the receptive fields from ganglion cells as well as the subunits, and then compare these subunits to the receptive fields of simultaneously recorded bipolar cells. So here are two ganglion cells, receptive fields, shown in a pixel by pixel fashion, and overlaid the fitted receptive field of a simultaneously recorded bipolar cell. And if we then compare this bipolar cell layout to the inferred subunits of these two ganglion cell, then we see that we often get a very nice match of individual bipolar cell receptive fields to individual subunits, giving us some confidence that what we get here in this analysis are actual bipolar cells that provide input to the ganglion cell. So we believe this is a very nice method of looking into the substructure of receptive fields. If you're interested, this may also be of interest to you that we have since then considerably improved the method. And this is thanks to this guy here, Sören Zapp in the lab, who has devised a much faster and more robust algorithm and is using this currently to get subunits also from primate retina, here from marmoset data, where you also find really nice layouts of subunits of individual ganglion cells. and using this to check, for example, if you have such a subunit layout, how this relates to other ganglion cells that are recorded simultaneously, and checking whether there are overlaps of subunits from different ganglion cells as an indication of common input received by different ganglion cells. But with this, I want to leave the subunits and quickly summarize that with this method of spike-triggered non-negative matrix factorization, we can computationally infer these subunits, and these inferred subunits match individual bipolar cells for us, at least so far in the salamander data. This all rests somehow on the assumption that the ganglion cells pool non-linearly over bipolar cells, but the bipolar cells really are mostly linear. So another question that we ask is whether that's actually true. So are bipolar cells then really linear and only providing nonlinear input to ganglion cells through their synaptic output? There's reasons to believe that they are or could be linear, essentially because they receive input from photoreceptors, which is modulated by horizontal cells. And in the retina, up to the stage of the bipolar cells, are essentially, except for some, maybe some exception in some species, there are no spikes. All these cells seem to be operating mostly on graded potential and continuously modulated transmitter release. So everything could be linear and is typically thought to be linear. But Helene Schreier already told you that she records from individual bipolar cells. When to check this with a single electrode recording, so poking a sharp microelectrode into the retina until she finds a bipolar cell, and then checking, for example, how these cells respond to spots of light on their receptive field, and comparing spots that are bright to spots that are dark or black, and checking whether responses are linear in the sense that you get reversed responses. And what she finds is that for some cells, this is more or less the case. So here for an off cell, you see this graded membrane potential inside the that is measured here by the sharp microelectrode. She gets a depolarization, and for a non-preferred contrast here, a white spot, she also gets a hyperpolarization in the cell that's dropping below baseline. So here 0 is corresponds to the resting potential or the baseline potential in the cell. But for other cells, this is not the case. So for example, cell 3 here shows strong rectification. You get a strong depolarization for a black spot, but essentially no response, no hyperpolarization by an opposite white spot. And then there are cells in between, of course. So it looks like that at the level of the membrane potential cells, bipolar cells can at least have a asymmetry between preferred and non-preferred contrast. And Helene then went on to check whether this nonlinearity also holds for stimuli that are continuous, such as spatial temporal white noise, by checking how the contrast signal of a cell inside this white noise stimulus relates to the membrane potential. So if you're familiar with the linear nonlinear model that I already mentioned, he here just measures the nonlinearity of this linear nonlinear model that provides information about how the filtered signal corresponds to the output, the membrane potential of the cell. If not, then you can think of this just by that for at every point of time, she checks whether the average contrast inside the receptive field of a given bipolar cell over the recent past, whether that was activating or not activating. And this average contrast inside the receptive field, we then call the filtered contrast. And then we can relate this filtered contrast to the actual membrane potential that was measured inside the cell. And here we see a similar picture as above. Here, some cells have a linear correspondence between the membrane potential and this average contrast over the recent past inside the receptive field, whereas others show a nonlinear, indicates rectification of the contrast signal here. And these are from the same cells here, so you can already see that a cell that is more linear here for the hyperpolarization is also more linear here for the white noise stimulus, and that holds true over a population of recorded cells. We can quantify the degree of nonlinearity here by computing an index, and we find that the nonlinearity, if we quantify this, that we get from the hyperpolarization. If that's large, then we also get a large measure of nonlinearity under the white noise stimulus, so that this seems to be a cell property, whether the cell is more linear or nonlinear. So here we see that the relationship between contrast inside the receptive field and the membrane potential of the cell can be nonlinear for some bipolar cells. Having established this, he then went on to ask whether we can also have nonlinear spatial integration by bipolar cell. And for this, she first measures responses to spots of light that are switching between black and white on the receptive field. And she sees that the cells respond. So here these cells all respond nicely to the black spot with a depolarization and then a hyperpolarization or decrease of the response when the white spot is presented. And then she, instead of the homogeneous spots, she also uses pattern spots that are, for example, separated into a white and a dark half, or here into quarters. So this is essentially the Enroth-Kugel-Robson type of experiment for bipolar cells, checking whether now you have a cancellation of these signals of white and dark, or whether you have responses to both reversals, which would then show also a frequency doubling. So let's see what these cells are doing. Cell 1 here, for example, essentially has a flat response. So it seems to not respond to these pattern spots, integrating linearly or canceling out the white and the dark activation of the receptive field. But other cells show a reliable response to these reversals. Here, for example, you see that the cell very strongly responds to both reversals and has a response that at twice the frequency here as for the homogeneous spots, and this frequency doubling here, these responses to both reversals for a pattern stimulus, like in the old X cell, Y cell work from Enroth, Kugel, and Robson, is an indication that there's nonlinear spatial integration in these cells, in these bipolar cells. This nonlinear spatial integration is actually also related or correlated to the nonlinearity that I've shown you in the previous slide, this nonlinear representation of contrast. So cells that are nonlinear in their representation of contrast under the white noise analysis from the previous slide also tend to be spatially nonlinear, as this cell here with frequency doubling for pattern spots. So this then raises the question whether, given that we have potential nonlinear contrast representation as well as nonlinear integration of the local contrast signals, shown here by the fact that there are local signals coming in that are nonlinearly transformed. So maybe we call this contrast signal here now the effective contrast or the integrated contrast signals. So we have these potential two stages of nonlinear processing. And the question is whether we then actually need the second stage here, or whether this nonlinear representation of contrast in the membrane potential is just inherited from this nonlinear integration here from the inputs that the bipolar cell receives, so that you would not need a second nonlinear stage here that would transform the effective contrast into a membrane potential. You can check whether this is the case, whether the single nonlinearity here is sufficient through a simple model, or in this model you can check it because this model makes a prediction about how the membrane potential for such a pattern spot, such a white-black spot, is related to the membrane potential of homogeneous spots of white and black. This turns out to be a fairly similar relationship that's shown here. The membrane potential that you should get for the pattern spot should be the sum of half the membrane potential for the white spot and half the membrane potential for the black spot. I'm not going to attempt to explain in detail how this relationship comes from this model, but the main ingredient here is that you can imagine if you have half a spot, the membrane potential that you measure for this half spot already contains all the nonlinearities that are part of this model. So if you then combine it with another half spot on the other side, the membrane potentials just add, because both these half spot activations have their the signals for these half spot activations already have their nonlinearities, and there's no further nonlinearity when you add them up and convert or predict in the membrane potential. So let's check whether this relationship here holds true. So here for 3 cells, we have responses to the reversal of a dark spot, bright spot. Let's subdivide this response into the response to the dark spot and the response to the bright spot, and then multiply them by a half and add them up in order to get a prediction for this pattern spot here, this black-white spot. So this prediction here is for one reversal, and then the same prediction here, of course, for this other spot, because these are essentially identical stimuli according to this model. So you have this response here shown twice for both reversals. And then we can check whether this prediction that comes from this model actually matches the measurements. And this is shown here. The measurements for the split spot actually match this prediction quite nicely, indicating, and this is actually true for most of the bipolar cells that we measure, we get a fairly nice match of the simple model to what we measure, indicating that indeed here we don't need any additional nonlinear transformation of the integrated signal into the membrane potential. All the nonlinearities in these bipolar cells can essentially be captured by nonlinear input that they are receiving locally inside the receptive field, which means that most likely the site of this nonlinear processing is before the soma, before signals are integrated, for example, on the dendrites or dendritic receptors of these bipolar cells, so fairly early in the bipolar cell processing chain. A lot more could be said about signal processing in these bipolar cells, but I want to get to one last point, so let me sum up this third part as well. So we've seen that bipolar cell nonlinearities exist in the shape that there is nonlinear spatial integration. What may this be good for? This may, for example, retain sensitivity to spatial detail. This may be particularly important for animals that have small eyes, where receptive fields get quickly large in terms of degrees of visual angle. If you want to be sensitive to a small object, you may not want to do too much spatial averaging and spatial cancellation. So that could make sense from a functional perspective. And then if you have this nonlinear spatial integration, we found that this also translates into a nonlinear representation of contrast, which is essentially inherited from this nonlinear spatial integration. And the potential site of this nonlinearity, we hypothesize maybe somewhere on the bipolar cell dendrites. Okay, finally, I want to ask, are there also other dimensions besides space that have interesting integration, linear and nonlinear integration? So let me show you an example here of a recording that was done by Muhammad Khani in the lab, who is analyzing a different type of integration. And here there are responses from 2 cells for 3 stimuli, the original stimuli, and here in the second column, the contrast reversed version of the stimulus. And if you look at this, and the fact that here cell 1 for the third stimulus is not responding and cell 2 is responding for both reversals, you may think this looks very much like what we've seen from Enroth, Kugel, and Robson, where for spatial gratings, you get responses for one reversal of the grating or the other reversal of the grating. And then at the sweet spot, where there's balanced excitation of the ganglion cell from black and white, some cells don't respond and others respond to both reversals. And it turns out that this analogy holds quite well also for what we are using here, but we are not using spatial gratings, but instead the stimuli that Muhammad has been presenting here are full-field color stimuli. So here, the first stimulus is a green flash of green light that's either turned off or turned, so increase in green light intensity or decrease in green light intensity over a gray background. And here the second stimulus is the same for UV light, either a decrease of UV or an increase. And here in the third row, you have the mixture of the two, increase in green and decrease in UV. I should add that the mouse retina has 2 types of cones, or rather 2 types of color sensitive opsins, and they are sensitive to green and UV, and therefore we here use this combination of UV and green. And what we see here, that the cell 1, apparently when you have the right combination of green on and UV off, there's a cancellation, you don't get a response, analogous to the X cells from Enloth, Kugel, and Robson. So this would be linear integration, but the cell 2 here, this actually seems to integrate color in a nonlinear fashion and then responds to both this original green on UV off stimulus as well as the contrast reversed version here. So this would be a sign of nonlinear chromatic integration. So having seen this, Mohammed devised a way to study this chromatic integration more systematically, and what he needs is a way to find the sweet spot where you have a balanced activation or balanced sensitivity to green and UV. And he does this very similarly to a spatial grating that you slide across a receptive field of a ganglion cell to find the balanced activation for both halves of the receptive field. He so to speak slides a contrast signal that goes from all green to all UV, and then with intermediate combinations, and then using also the contrast reversed version of the set. And then you can look at the responses for ganglion cells, and for example, check as you go along this curve, this set of stimuli here, how the responses decrease from being activated by green light to potentially going below baseline activity level. And compare this to the responses of the contrast reversed stimuli that are shown here, increases in activity, and then you find the sweet spot where a cell is equally sensitive to one stimulus and the reverse stimulus at the point where these two curves cross. And then you can check for this crossing point here, is there activation or no activation? In this particular example, we see that there is only baseline activity, no activation, so this would correspond to a linear cell. But for other cells, we see that this crossing point here is above 0. There is actual activity here at this crossing point corresponding to nonlinear chromatic integration and responses to both contrast reversed versions of color combinations. Let me show you a few more examples of responses at this crossing point here. This is a linear cell with no response for the right combination of green and UV. This is a response similar to what we have seen here, increased responses to both contrast reverse stimuli. In this case, this is an off cell, but then we also find a third type of response here for an on cell, where at this balance point, we actually don't see a peak in activity, but we see a decrease of activity below the baseline activity. And this interesting pattern here, that for some cells we see an increase of activity and for others we see a decreased activity, is actually systematic for on cells and off cells. So at this balance point where UV and green stimulation are equally effective, essentially if cells are nonlinear, then all off as well as on-off cells show this increased activity, and all on cells, if they are nonlinear, show a decrease in activity. That's very systematic and probably tells us something about the circuitry of how this nonlinear chromatic integration is established by the retina or comes about. So just a few quick words about where we believe this nonlinear chromatic integration comes from. The first thing that you may think is, well, we already know that for spatial integration, we have linear and nonlinear chromatic integration. And this comes from bipolar cells, and maybe it's just the fact that some ganglion cells are always linear and some are always nonlinear, regardless of whether they integrate or whether the experimenter looks at integration over space or over chromatic channels. So we checked this, but we found that it's not the case. So the nonlinear integration under chromatic stimulus is different from the nonlinearity that you get under spatial integration, essentially because we have cells with all, or we have cells for all combinations of linear and nonlinear chromatic integration and linear and nonlinear spatial integration. So here, for example, is a cell that is chromatically linear. The crossing point is here at baseline, but the cell responds very strongly to reversing gratings, fine spatial gratings, indicating spatial nonlinearity. And here we have a cell that's chromatically nonlinear with the crossing responses to a reversing grating. And we find that these two properties are just not correlated. They don't have much to do with each other. And then we also find that this nonlinear spatial, nonlinear chromatic integration seems to come from the surround, from the receptive field surround of the cells. That seems to be important for these cells, because we do find it when we use full field chromatic stimuli, as shown here for an off cell and non cell, crossing point different from baseline. But when we do the same experiment with small spots that we place on the screen at different locations and cover the entire screen with spots presented at different times, and then for each cell find the one spot that matches the receptive field location of that cell well, then we see that this chromatic nonlinearity disappears. Now these crossing points here drop down to baseline activity. We don't get an activation at the crossing point anymore, indicating that for local stimulation, we now have linear chromatic integration. So the surround seems to be important for this. Finally, let me just make another remark about this chromatic integration that we find. It's that nonlinear cells are not homogeneous. The cells are on the retina. So this is an image here of a recording from an entire retina and recorded receptive fields, the dorsal side of the retina and the ventral side of the retina. And if for each cell we check whether it's linear or nonlinear in chromatic integration and relate this to the position of the cell on the retina, then we find that the disc here is shown from the distance to the midline of the retina, that in the dorsal retina, we mostly find linear cells here. Those are the blue spots here that are here on the dorsal side, whereas the nonlinear cells here shown in orange are primarily on the ventral side of the retina. So whether a cell is chromatically linear or nonlinear seems to depend strongly on where it is on the retina. We also find some cells that don't integrate UV and green light, that are exclusively sensitive to UV cells, and those are also mostly in the ventral side of the retina. Okay, finally, some speculation about a potential function. This we study by setting up a model of linear and nonlinear chromatic integration, where you take two signals from green and UV light, and either you just add them to get an activation of a cell, or you first rectify them and then add them up, which is a simple phenomenological model for the type of responses that we've here found in the off-type nonlinear chromatic cells, because this allows the cell to respond to opposing contrast in green and UV. So for such a stimulus here, you would get a response in these nonlinear cells, but no response in the linear cells. And then we took images from a database that photographed images through the same images through both a UV filter and a green filter, so where we have the two color channels, and then checked how these model cells respond to the color information in these images. And here I just show you the difference depending on the location in the image, where the nonlinear integration gives a different response than the linear integration. And it turns out that this nonlinearity is important, gives a different response than the linear model, approximately here at the line that divides the foreground from the background sky illumination, so the skyline, so to speak. This is where you can have this opposing contrast of green and UV light, probably because of difference in reflectance properties of the foreground. And therefore, you see here that the nonlinear cells pick this up and respond strongly, more strongly than linear cells, giving a potential channel of finding the skyline by comparing responses between chromatically linear and chromatically nonlinear cells. Okay, let me sum up this last part as well. So we find that chromatic integration is another interesting dimension where you find also linear and nonlinear integration, very similar to spatial integration. This nonlinear chromatic integration appears to depend on the surround, on activation of the receptive field surround, and it's primarily found in cells in the ventral retina of the mouse retina. So let me just very quickly recap the 4 parts of the talk. We found nonlinear receptive fields in the mouse retina that are relevant for natural stimuli in many cells. I've shown you that you can get the subunits of receptive fields through computational techniques. I've also shown you that in the salamander retina, at least bipolar cells can also be nonlinear, show nonlinear spatial integration. And finally, that there can also be chromatic nonlinear integration, where different color channels are combined in a nonlinear fashion. And with this, I would like to thank the people involved, the people in the lab. I've mentioned the people that have done the particular parts that I've talked about here. I should also mention that this spike-triggered non-negative matrix factorization was developed in collaboration with Stefano Panzeri and Arno Onken from the Italian Institute of Technology. And finally, I would like to acknowledge the funding sources that have supported us, the ERC and the 7th Framework Program from the European Union, as well as the German Research Foundation through a collaborative research center. And with this, I'm at the end. I'm very much looking forward to questions and an online discussion. Thank you.
Hopefully I'm audible. Can you hear me, Tim?
I can hear you.
Oh yeah, well, that should be enough, shouldn't it? Thank you. There was actually a lot more interest in things than even I expected, and I invited you. So no, so this was great. We have a great deal of questions. So shall we start right now? And
sure, absolutely.
Yeah, so I'll just remind you to look out what will be happening in the chat. So we'll be posting a link to this very room, to this very Zoom room in which we two find ourselves, so that you can join the discussion live a little bit later. Right, so questions. There are plenty of those. I think I will group two together. So there's a question by George Kafetsis and a question by a certain Marcus Meister that sound a bit similar. So probably minor, but given that you talked at the start about resolution in and out of focus sort of images, how does the stimulus reach the retina on the MEA physiologically through RGCs? And then Marcus Meister also asks something similar in spirit regarding whether these effects are important for natural vision. Were the natural images scaled and filtered to emulate what would actually appear on the retina of a mouse or a marmoset?
Yeah, good questions. So in the experiments, we actually simply present the stimulus on a computer screen, and that is projected onto the retina from the photoreceptor side. So in some sense, from the wrong side of the retina, which may indicate that there may be less blurring, for example, of an image. This is something that we don't take into account at the moment, which is definitely an interesting follow-up question. At the moment, we simply focus on the fact that if you compare, let's say, natural images that have objects as well as contours, and compare this to see whether this makes a difference to the typically used artificial stimuli like gratings and on-off stimuli, and see whether there's a relevance on this level. But a good next step that we haven't done yet is to match this better by, for example, trying to match the blurring that occurs by the optics of, let's say, a mouse or a salamander eye, and the passing of light through the retina. But at the moment, we haven't done this yet, no.
Right. So thank you for the response. I just remind everyone that you can keep asking questions as long as Tim is alive and willing to respond. Somewhat more technical, perhaps. So Brent Young is asking, so it is very interesting that there is such a strong on response in some cells with a uniform gray field. How different is the light intensity of these natural image grays versus the solid field you used before? That was at the beginning of the talk.
Yeah, so I guess strong on response may refer here to a response at the image onset. I'm not sure whether I understand this correctly. So the way the experiment is done, that there is a intermediate gray illumination between images, and then the images over the entire image have the same mean luminance. So in that sense, there's equal chance that the ganglion cell sees an increase or a decrease of light intensity. But of course, since each individual cell only sees a small part of the image, it's, let's say, unlikely that it sees the same average gray inside the receptive field than was the background illumination. That can be. And then if the cell is a linear cell, the cell should not respond, right? Because it would see an average 0 activation. But a nonlinear cell can then, of course, still respond if there's structure inside this part of the image, so that it sees decreases as well as increases in light intensity. And that can be considerable. So even for an image patch where the net activation, the average luminance signal is essentially 0, is the same as the background gray light intensity, you can have strong responses just because there's spatial structure inside the image.
Right, I think that answers it. So we have another technical question about non-negative matrix factorization and the penalties that you've used, what loss and regularizations you have used, and whether you expect different results, I guess qualitatively different results, if you change the losses and regularizations.
Yes, that's a good question from someone, I guess, who knows what he or she is talking about. So in the original paper, we used regularization that we essentially took out of the box, and it was an L1 regularization on these subunits, which helped to make them sparse. And if we don't do this, they are more blurred, let's say. And we essentially took it out of the box without tuning it much, because it worked well in the sense that it gave us consistent results, that if we wiggled it a little bit, responses didn't change much. And if we increased the regularization too much, we would get individual pixels. And if we reduced it too much, we don't get nice subunits. So in sometimes that can be considered a weakness, and therefore we've worked on this since then. And in the new version that I hinted to, we now have a version that works without regularization. So where we don't need the regularization and the marmoset receptive fields that I've shown you, you get without any sparsity prior. So any regularization, any L1 regularization. But it probably will take still a few weeks before this is really finished and hopefully out sometime soon, but that's coming up. So you can work on this and improve on this. And we've also looked at other ways of checking the regularization, so you can use regularization and find ways of gauging the right regularization level. For example, by checking that if you use different subsets of the data or different initialization conditions, you get particularly stable solutions. And that seems to be a useful way of doing something like cross-validation to find good tuning of regularization here.
Well, that sounds particularly exciting for some people, I guess, well, myself included. But I think that there are more questions that are sort of connected to this and are connected to the later bit of your talk. So Whaley exclaims that it was a great talk and asks whether you have found any chromatic subunits within the RGC receptive fields. If so, what do they look like? And so somewhat connected, maybe these two group. Maximilian Josh is asking that since UV light also stimulates M opsins, wouldn't that explain the linearity in the dorsal retina?
Mm-hmm. Yeah, good questions. So first about the subunits. So the short answer is no, we have not, because we have not looked sufficiently. So far, we have only analyzed data where we used monochromatic stimuli, so just grayscale, and therefore we have no information in about chromatic aspects of the subunits. Interestingly, we do have data where we just haven't gotten around to analyze this. So in some recordings, we also use chromatic stimuli, so 2 color channels, high spatial acuity stimulation, long recordings. This becomes a bit more difficult because you double the number of parameters essentially by taking 2 color channels. So this becomes a bit more involved, but we may have an answer there sometime in the future. Too many things to analyze. The second question, stimulation of M opsins. So that's true. So the UV light does activate M opsins. A thing that I skipped over is that whenever I talk about UV and green contrast, what I actually mean is M opsin and S opsin contrast, because we do all these experiments with cone isolating or opsin isolating stimuli. So really, when we use UV light, we at the same time or talk about UV contrast, we at the same time modulate the green contrast in such a way that that the M opsins are not or should not be activated. Otherwise, this could be contributing to a linearization effect. That's true.
Right. So we have some more questions. One is from a certain Leon Lagnado. I don't know who that might be. So he mentioned spatial nonlinearities in bipolar cells. Are you, Tim, thinking about variations in the density of photoreceptor inputs to passive dendrites, or active conductances, or both, or neither, or neither?
I guess the question is what we're considering as a mechanism. Is that the question? Yeah.
I mean, you can look at the text of the question. I haven't paraphrased it too much.
I have to.
So it does mention.
I'm thinking about. Okay, yeah, now I got it.
In essence, there are probably several mechanisms there.
Yeah. So I guess both. So at the moment, what we can say is that it's likely that something is happening at the dendrites of the bipolar cells, because it seems to be before spatial integration, and it doesn't seem to be happening at the level of the photoreceptor per se, for 2 reasons. Because we also record from photoreceptors and we find fairly linear contrast representation in photoreceptors. So at least at the level of membrane potential. And one could also say if it's the photoreceptors, then probably it should be all bipolar cells that have the same linear or nonlinear property. So that's the cheap argument, so to speak, to say it's not photoreceptors per se. That could still mean it's somehow in the synapse between photoreceptors and dendrites, and maybe more likely postsynaptically. And yes, those 2 aspects are then the primary candidates that we would consider, that it's somewhere, so to speak, at the synapse, maybe on the postsynaptic side, how the dendritic receptors, what they do with the photoreceptor neurotransmitter signal, whether they locally transform this in a nonlinear fashion. And the second aspect could be active channels in the dendrites, which are known to exist. So that's a good candidate as well. And at the moment, we can distinguish between the 2.
Mm-hmm. Oh, we have more and more questions. Can you carry on? We have many more questions coming in.
Oh yeah, I can carry on.
Well, that's great. Right, so we have several interesting ones. I'll go with Anton Nikolaev. Can the noise subunits not be noise but reflect the activity of amacrine cells?
Mm-hmm. Yeah, I see the point. I mean, most of these, if you looked at these noise subunits in the spike-triggered non-negative matrix factorization closely, they seem to have their pixels mainly in the surround. So one could say, so maybe they have something to do with the surround. Maybe they are, a contribution of amacrine cells. So the reason why I say they are mostly noise is if you look how these filters, if you take them as stimulus filters, are related to the response of the ganglion cell, you essentially see a mostly flat line. So if you filter the stimulus with one of these filters that I call noise, and then compute, let's say, the nonlinearity for this filter, or correlate the activation of this filter with the response of the ganglion cell, then it's really markedly flat, except for something that is most likely noise, and therefore we interpret them as noise signals. So the thing is that we do this analysis with a fixed set of filters that we typically use in a way, try to tune in a way that it's larger than the number of subunits that we have, because this allows us to capture some of the noise that's in there, just because the stimuli that we use are Gaussian distributed pixel values. So there's never, the exact stimulus that drives a subunit. There's always variations, and turns out that it's easier to fit the subunits if you provide, so to speak, a trash can where the variations in the pixel intensities can be stored, and this is essentially what these other subunits then do. So if we don't allow for these noise subunits, then also the fit of the actual subunits gets worse.
And that makes sense. So we have more questions. So one, I don't know if this was answered during the presentation. Right, so it came somewhere in the midst of your talk. So these computations, Etienne Roche is saying that these computations are interesting for the processing of features in natural scenes. To what extent do you think they would be involved in processing exempli gratia, symmetry, edge detection, etc.? Do you think this was answered? Or do you want to?
Probably not. I mean, I can say a few things. So, so the interest in nonlinear integration, let's say, partly comes from interest in the circuitry. How are signals from bipolar cells relate to ganglion cells? Is that linear or nonlinear? That tells you something about the synapses, maybe. It's also triggered by the idea that we want good models, and we want to know what we have to integrate in these models in order to get good predictions for ganglion cells, both for, as a framework for checking, do we understand what the retina is doing? Maybe in the future also to build prosthetics or whatever. Good models are always useful, right? So that's an interest. The second interest is that most likely, I mean, we know that some cells in the retina are doing specific computations. Some detect the direction of motion, other maybe the looming stimulus or whatever. And these computations are really only possible if you allow the cell to do interesting things inside its receptive field. And these interesting things mean that somehow the cell has to combine signals over its receptive field in a nonlinear fashion. Otherwise, it cannot really do anything, let's say, interesting, specific, that goes beyond, contrast filtering. I mean, in some sense, interesting is here defined as being nonlinear, right? So in that sense, if you're looking for a computation like detecting direction of motion, maybe, or maybe edge detection could be a similar mechanism that's coming in a particularly robust fashion, then it's likely that this will involve nonlinear mechanisms. So I would say yes. I mean, all these nonlinearities that we see are likely something like, maybe you can call it the scaffold or the layout on which nonlinear integration or on which specific computations can be built. Right? You can then combine these non-linearly extracted elements in a certain way in order to extract some information. What that is, we probably need to do a little bit more work, right? Just by saying that a cell is nonlinear doesn't tell us what computation it may be doing, right? But you can then go on and ask what kind of nonlinearity is there. And in the talk, we've seen two different types in a way, right? Some cells that prefer contrast inside the receptive field, and some cells that prefer homogeneous stimulation. And that may give you some idea about the function. But you can go further, right? You can not only ask, is the cell sensitive to spatial contrast, but does it use the spatial contrast in, I don't know, a rectified fashion, or does it compute the square of the contrast, and how does it combine these signals? And that may, and there are examples in the literature, give you an idea of what sort of computation is then implemented by these nonlinear circuits. I have no idea whether that answered the question.
Certainly gave me more questions to ask, but right. So I'm being told to move to the conversation to Zoom. Although we have more questions that were asked in the chat room. So what I will now try to do is I will try to interact with my colleagues and ask them where the link to the chat room is, because it should already be in the chat. But so please, colleagues, someone do the feed and post the link to this chat room. Right, and meanwhile, I will ask you maybe one more question.
Sure.
So have sorry, this is this is color related, so we will have that. So Marcus Howlett is asking, how well do opsin isolate and stimuli isolate cone output, considering horizontal cells are spectrally broadly more broad than cones?
Yeah, so I guess there's a general question of how well cone isolating stimuli are really cone isolating, and therefore maybe the safer term here to use would be opsin isolating rather than cone isolating, right? Because given from feedback from horizontal cells, you know, the cone output signal may not represent
the general question of how well
opsins inside the I'm hearing myself now in the background. That's a bit irritating. Never mind. I'll just try to go on. So the cone output signal may not reflect the opsin inside the cone. And the second aspect is that also in the mouse we have a lot of co-expression of opsins, and therefore also we cannot isolate these cones, obviously, but we can try to isolate the opsins. Yeah, so in that sense, I would more go with opsin isolating stimuli. That should work fairly well because this is sort of well established in the literature. And from our measurements, it also looks like it's working quite nicely, given the fact that we see a good correspondence with how cells in the ventral and dorsal retina are activated by our opsin isolating stimuli, and from the fact that we see UV sensitive cells that are not responding to green light, for example, or M-opsin activation, I should say.
I guess I will nearly disappear into the background now. I know that there will be plenty of questions, at least with these two around.
Okay, hi everyone.
Do enable yourself to speak.
That was great. I really enjoyed that. Lots of new stuff.
Yeah, mostly all new.
And turns out that it's like jazz, right? Retina's like jazz. Eventually we all end up with color.
Some time ago, I heard someone, eventually you all end up with direction selectivity.
Well, no, color.
A bit of both, yeah. Color.
Yes, it's on a stepping stone.
Color is the new black. Yeah, color is certainly interesting.
So I don't know if you know the study in detail. So some time ago we showed that UV cones in the mouse are very nonlinear in the contrast response, whereas the green ones are linear-ish. So do you think that's driving some of these effects you see?
It could be, yeah, absolutely. That's a potential candidate for adding nonlinearities. I guess both nonlinearity as well as potentially, although there was not mouse, it could be a potential contribution to the nonlinear bipolar cells that we found in the salamander, if potentially something similar is happening. Yeah, for the chromatic nonlinearities, I guess, I mean, our primary candidate for a mechanism probably goes through amacrine cells because of this connection that we have via the receptive field surround, because we really see that we need receptive field surround activation in order to trigger these nonlinearities. I've shown you these spot stimuli. So for local spots, the nonlinearity disappears, but we can also make it disappear by certain inhibitory blockers. So I would guess that the nonlinearities from the photoreceptors should be more robust to blocking inhibition. I guess on the level of the type of nonlinearities that we see here on the level of ganglion cells, part of the nonlinearities that come from photoreceptors could be washed out a little bit by integration by bipolar cells. If you have chromatically mixed bipolar cells, potentially even, more or less linear. I would have to think this through. What expectation what contribution you should expect from.
No, I guess the bipolar cells are funny in that way, right? Because they get these massive inputs from amacrine cells, which you may or may not be seeing the way they are when you poke them in the soma, right? So it's an enigma.
Yeah, absolutely. So I mean, in a way, we expect that in the salamander data, the nonlinearities that we've seen in the bipolar cells, we would assume that these nonlinearities already come before potential contributions from amacrine cells, because they are at the soma. Because we may not be able to, because I guess we don't expect to see too much of the amacrine cell input that goes into the terminal.
Yeah.
But we can rule this out. This could be a contribution, right? The cells are not that big. Something could be back propagating.
Yeah.
So one argument that we have there is that we see the same nonlinearities in the bipolar cells, regardless of whether we use small spot and large spots. And as we've seen from recent literature about bipolar cells, right? At least the inhibitory contributions to bipolar cell activation seem to depend or are strongly modulated by whether you have a small spot or large spot. And therefore we believe what we there see, because it's also there for small spots, doesn't depend on amacrine cell activation and back propagating signals. Activation seems to depend.
I give way.
Okay, there seems to have been a question, but then.
Yeah, so please remember to enable your mic before you start talking, right? There's a question from Jennifer to everyone. One thing that always confounds me is how to think about how such specific spatial information encoded by these cell types would translate to a rapidly moving eye. How do you think about how this information would look different to animals with rapid eye head movements versus those without such movements like the salamander? It's a question mostly to Tim, apparently.
Yeah, I think this is an important question and probably a strongly under-investigated question that I find also particularly interesting, and therefore we have been looking a little bit into this. I mean, a hand-waving argument is that at least in the first part of the talk, I showed you responses to more or less briefly flashed images, 200 millisecond flashes of images.